Okay, so hello everyone. Thank you so much for coming today. For those of you who are either new to DMICE or who are from outside of DMICE and may not be familiar with the style of this talk, my symposium presentation today will be a sort of research in progress talk and an opportunity to tell you about the research I'm really interested in before I actually give my dissertation, before I propose my dissertation project. Okay. So first I'll present the question, or actually a set of questions that got me started, and then most of the talk I will focus on prior work that other researchers have done to address some aspect of my original question. And then lastly, I'm going to share with you some gaps in the field that I might be able to tackle in my dissertation. It would be immensely helpful to me to hear It would be immensely helpful for me, especially when it comes to the research gaps, to hear what does or does not resonate with you and why. Okay. So obesity is expensive, uncomfortable, and deadly. So along the x-axis on this graph is body mass index with relative risk of death on the y-axis. And as you can see for the curves for both men and women, the relative risk of death increases substantially with a BMI above 30. And as you can see from the arrows across the top, this comprises nearly a third of the U.S. population. Okay, so naively I started with this very simple model of obesity. Greater food intake and less physical activity lead to obesity. I'm not here to argue against the first law of thermodynamics, and while I hope everyone here can appreciate this very simple model, working model of obesity, that it's technically correct, I'm going to propose to, is it really useful? And I would argue that it's not. If it were, everyone would eat healthy food and move and wouldn't gain weight. So despite being a very ardent champion of Occam's razor, this model is too simple. Which leads me to my motivating question for today, which is, why exactly isn't this model of obesity more useful? So to answer this question, I first try to address what combination of environmental exposures and common genetic variants do we think cause obesity in humans? And in the process of answering this question, I also began to wonder whether the reward system might be a useful theoretical construct to learn more about the cause of obesity. It is my hope that the answers to these questions might lead to rational prevention and treatment strategies. Okay, so I know it seems like very simple, but I think it's always good to start with definitions. So what is obesity and how is it measured? So overweight and obesity are defined as abnormal or excessive fat accumulation that may impair health, and this definition comes from the World Health Organization. The body mass index, or BMI, is a simple index of weight for height that is commonly used to classify overweight and obesity in adults. However, BMI, which is defined as kilograms over meters squared, is just one of many measures of obesity. BMI, along with the other measures of weight relative to height on the top bar, all make the underlying assumption that excess body mass is from fat. So the remaining measures on this chart do not make this assumption and instead attempt to quantify a person's fat mass separate from their fat-free mass. So while, and then the density-based methods along the bottom, especially with the underwater weighing, that's considered the gold standard when it comes to measuring obesity. So depending on the researcher's primary question, he or she will use a method that best estimates the amount and location of fat or adipose tissue on the body. And oftentimes, BMI is sufficient and a practical measure of obesity for studying health, especially at the population level. Okay, so back to my naive model. If I want to better understand the cause of obesity, I then wanted to say, what useful complexity is missing from this model? So first, I wanted to propose that we add some important environmental exposures. So namely, individuals that have access to food. Namely, individuals must have access to food in order to actually eat it. So to understand how food access affects food intake, I want to show you two studies that I found really interesting and helpful to address how food access really does influence food intake. So the The first study attempts to predict actual foods people eat given a cost constraint. And the second uses a measure of the level of poverty in the area where the study participants actually live to predict BMI. So the first study demonstrates that cost adversely affects food intake. So how did they come to this conclusion? So to conduct the study, the authors collected food intake data from 1,100 people through a dietitian-administered survey in Paris, it happened to be. And the authors simulated how a reduction in total food budget, shown on the x-axis here as a cost constraint in euros per joule, would affect food intake based on the actual price of different foods using a retail food price database. So to be precise, given the method here, the objective was to design a diet that most closely resembled the mean diet observed in the population while keeping the total energy intake constant. So it's basically like trying to keep, say, what would a rational individual do given that their budget is decreased? So from this simulation, they found that a reduction in total food budget predicted a smaller proportion of energy from fruits, vegetables, meat, dairy products, and an increase in energy from cereals, sweets, and added fats. Then they quantified using a food composition database how each lower cost diet would affect energy density, percent energy from fat, and vitamin C content. So as you can see from the three curves, so percent energy from fat, as you go down and reduce the total budget, the percent from fat stays actually pretty constant, but the energy density increases substantially and the amount of vitamin C decreases substantially, and that for each of the diets depending on the total cost. And so they chose vitamin C mostly just because that's a good measure of micronutrient content of the diet. And then energy density is another well-known, a marker used in nutrition research for whether or not that's highly associated with earlier death. So, yeah. Okay. So, and these results are important. Oh, yeah. Okay. So, so the next study I want to tell you about shows that genetic risk for obesity disproportionately affects people living in areas of poverty. So this study was also really interesting. So along the x-axis here is a genetic risk score for higher BMI, where people with a score of 10 have the highest risk, have a collection of genetic variants with the highest risk for obesity. And the people with a score of 1 have a collection of genetic variants with the lowest risk for obesity. And then actual BMI is plotted along the y-axis. So as expected, people with a higher genetic risk score for obesity have a higher BMI, like the positive trend that you see, very obvious. But the important thing to note is that the magnitude of the effect is much worse for people living in areas of poverty. So shown here, by the black circles, these people have, these are SEP stands for socioeconomic position. And to determine whether people actually lived in an area of poverty, the authors mapped each individual's postal code to where they were from. And that was done using the postal codes. This study was done in the UK. So they collected this information. And the level of poverty in each postal code was determined using what's called a meaningful measure of poverty, which was the Townsend Deprivation Index, which measures the amount of unemployment, non-car ownership, non-home ownership, and household overcrowding in each postal code. Okay. Okay, so I really liked that study. However, I found this paragraph in the method section a little confusing, and I'm going to read it to you. So the obesogenic environment refers to an environment that promotes gaining weight and that is not conducive to weight loss. Here we use the term environment to refer to any variable that describes a component to obesity that is not genetic variation. Many of these measures are likely to be a complex mixture of environment and behavior. Okay, so the reason I found this a little confusing is because we know behavior and environment are different things, and there is a tendency to sort of lump them together. So perhaps we can improve our model of obesity by just separating them out. Behaviors, yes. Okay, So I want to take you through now some important landmark studies. So I feel pretty happy about the adding the food access. So I want to take you through some important landmark studies that show how genetics affects obesity. Okay, so this is a question we're asking. So the first thing to recognize is that some people are obese due to a mutation in a single gene. A trait is, just for some definitions in case everyone in the room doesn't remember, a trait is monogenic or Mendelian if a genetic variant is both necessary and sufficient for the trait to be expressed, given the normal range of human genetic and environmental background, variation in human genetic and environmental backgrounds. So how do we know that obesity is monogenic in some people? So first we found through patient family histories of obesity that the inheritance patterns looked Mendelian. And so by Mendelian, I mean that they followed one of the 5 patterns that you probably learned about in high school. So autosomal recessive or dominant, X-linked recessive or dominant, or Y-linked. So over time, using animal models of obesity that have Mendelian inheritance patterns, we were able to figure out what these genes actually are through positional cloning. And we then confirmed that these same gene mutations, characterized in mice were also in real families with monogenic obesity using linkage analysis. Okay, so Zheng et al. Is the author who first published the positional cloning to map the first mouse obesity gene in 1994, which codes for the hormone leptin, which is right here. And leptin is released from the adipose tissue in proportion to the amount of fat mass, which then acts on the leptin receptor here in the hypothalamus and to regulate food intake. So a number of other groups mapped each of the genes involved in what is now known as the leptin-melanocortin signaling pathway. And importantly, subsequent linkage analysis of severely obese children demonstrated that genetic variants in the melanocortin-4 receptor, which is up in the blue circles, is the most prevalent form of human monogenic obesity. And you can see from this sequencing panel that the genes we test for in patients with suspected monogenic obesity today encode important proteins in the leptin-melanocortin pathway, and those are here. Okay, so it's also very interesting to note that BMI in a general population, in this case of about 500,000 people, is associated with more common genetic variants in genes that are part of the leptin-melanocortin pathway. So we can see this relationship in the figure here. So BMI categories are across the bottom on the x-axis, and along the y-axis is the proportion of people in the population, the 500,000 people, with 2 or more common genetic variants in the melanocortin pathway. And you can clearly see that there are way more obese people with common genetic variants in this pathway than in lower BMI categories. So this is way higher than this. Okay, so this suggests that the genetics that contribute to BMI, even in people without Mendelian inheritance patterns for obesity, are important. Okay, so we think that common genetic variants contribute to obesity in patients without Mendelian inheritance patterns. In genetic epidemiology parlance, we would call obesity a complex trait. So what exactly is a complex trait? It's a trait where the cause in a single individual could be due to a single genetic variant, many genetic variants, entirely environmental, or some combination of genetic variants and environmental exposures. So how do we know that obesity is likely caused by a mix of genetic and environmental exposures in a population? So to do that, we first ask the question, is there evidence that the trait is at least partly genetic in a larger population? And if so, where are these genetic variants actually located? So I'm going to do a because I think it's fun and helpful to understand. So twin studies consistently show that approximately 80% of the variation in BMI can be explained by a variation in genetics. This table shows the correlation in height, weight, and BMI, here, both at baseline and at 25-year follow-up. And here at the first time point, the people were like around 20 years old. And the estimated hereditary in each of these two columns show the intra-pair correlation between the first one, this is monozygotic twins, so people who share all of their genetics, and dizygotic twins, which are basically like siblings, but should theoretically be assumed to be sharing half of their genes. And then G is a calculation of heritability based on, like I said, based on this assumption that dizygotic twins share half their DNA and monozygotic twins share all of their DNA. So I think it's like interesting just to like look at this, right? Like you see body mass index is here at around like 20 years old and 84% at 25 years later. No, these are just twins. Yep, so very good question. So the next study. So adoption studies show the importance of genetics and not the environment on obesity. It's a little bit of a strong conclusion that the authors made, but let's roll with it for a moment. So adoption studies in general are considered the gold standard for disentangling the genetics and environmental factors. So how does an adoption study work? So researchers identify a cohort of people who are adopted, and then they simply compare their BMI to the BMI of both their biological parents and their adoptive parents. So in the figure above, the weight class of the adoptees is here and here. And then the body mass index of the parents, either biological parents or adoptive parents, are here. So what I hope is fairly obvious is that the correlation in body fat and body mass is a pretty strong correlation for the adoptees and their biological parents, and that relationship is not strong for the adoptees and their adoptive parents. Okay. So I don't think this study rules out a role for the environment on obesity. It does demonstrate that the genetics is surely important. Okay. So in order to find out where these genetic variants are located, researchers started looking for chromosome regions associated with obesity. And they did so by using genome-wide linkage analysis. That's what they did first. And the finding from these studies mostly re-identified regions that we sort of already knew from monogenic obesity, at least from my background research. And in this study in particular, genetic markers in the region containing the melanocortin receptor were shown to be associated with BMI and a % body fat measured by biomedical electrical impedance analysis. So this region here, I should put on, which when they actually reported it the first time around, they just said this region of the chromosome, but later it was shown that that's where the melanocortin receptor was. So. Okay, so with the sequencing of the human genome complete, researchers were next able to ask if there were any common genetic variants associated with obesity genome-wide. So the first studies identified fat mass and obesity-associated or FTO gene. So while people were sort of underwhelmed at the time because there was not any clear explanation as to why FTO was associated with obesity, it was actually pretty cool since it suggested that there are other genetic mechanisms responsible for obesity in humans aside from the leptin melanocortin pathway. So since 2007, genome-wide association studies for BMI, waist-to-hip ratio, and lots of other adiposity traits have identified more than 300 single nucleotide polymorphisms related to obesity. And I thought this was an amazing Venn diagram. But you can see that a number of SNPs that are located close to genes and associated with, they have BMI here is in the middle, body fat, waist-to-hip ratio, visceral adiposity, extreme obesity, birth weight even is sort of a little different. So that's sort of the state of the field for genome-wide association studies in obesity. Okay, so that was sort of long, but I think kind of a necessary tangent, so we can all be on the same page about how genetics contributes to obesity. However, we know that all of these do we know that all of these genetic variants affect obesity directly? So what if these variants actually affect the behaviors that lead to obesity, like this? I think this would be important to know if we have any hope of preventing or treating obesity. Genetics associated with variation in physical activities outside of the scope of my talk today, but probably very interesting. Okay. So the reason I became really interested in looking at this and looking at the genetics of food intake rather than obesity alone was due to another interesting perspective published by a group looking at a separate complex trait, so specifically lung cancer. And this group was interested in understanding the way genetics and behavior contribute to lung cancer. So when they started, they knew that smoking causes lung cancer, and they also knew that specific genetic variants labeled here from a previous genome-wide association study were associated with lung cancer. But upon further investigation to the known mechanisms of these genes, where these variants were located, the authors reasoned that the most likely explanation for their association with lung cancer was actually due to their effect on smoking behavior. So does anyone have any guesses as to what these variants are? So they code for the nicotine acetylcholine receptor subunits. So they're highly associated with people's response to nicotine. So while it's technically still possible for these variants to exert an independent effect on the risk for lung cancer, I hope you can appreciate that most, if not all of the genetic variation of these genes, probably exert their effect on lung cancer through smoking behavior. So, like this. So I thought, could this framework apply to obesity and food intake genetics? Okay, so to evaluate whether this might work, I first needed to look at the literature to see if food intake is at least partly genetic, and if any of these genes variants have been identified. Sort of the same questions that I asked for obesity. So while there's been no gold standard adoption study of food intake to date that I could find, twin studies on food intake do indicate that the trait is at least partly genetic. But it should be noted that the heritability, as seen from this summary table, are definitely less than the heritability of obesity. So here, like the longest, probably most rigorous ones with the most N were like maybe 0.5. But that would be optimistic. Okay. So what about the locations of genetic variants associated with food intake? So genome-wide linkage studies, as with obesity, identified chromosomal regions that are part of the leptin-melanocortin pathway. However, few genome-wide association studies have been completed. However, the very few number of genome-wide association studies that have been completed on food intake have shown some novel variants. So I put these two together because they basically used the same data sets and showed the same results. So that's why they're like the same thing. And then these two were two other studies and returned. So again, FTO we already knew from obesity. FGF21 was a new finding. And then these two genes were also associated with, this was a confectionery intake was like a survey, and fat intake for the ADIPQ. Okay. Okay, and then, so when I first started looking at this, I was like, oh, it would be so good to like make sure this is true. And nobody had done it yet in the UK Biobank data set. So I was like, oh, I should do that. And then that came out last month. So someone else did that. That's so good. I guess it's good I don't have to do it. But it's good that the results replicated. So both FTO and FGF21 were replicated in the larger cohort. And they also found 2 others that I didn't put up here, but I just wanted to show you that like those 2 were like found again in another 3 separate populations that were included in this study. What was sort of confusing, or not confusing, but just interesting, is that like this issue of like looking at different macronutrients and how these variants are sort of associated with a mixed match of which macronutrient. So humans get their energy from fat, carbohydrate, protein, and also alcohol. And these macronutrient intakes as a percent of total energy are correlated to some degree because we're eating foods that contain somewhat conserved relative amounts of these macronutrients. So since we're talking about genetics, I wondered, is there any evolutionary theory that might help explain why genetic variation would contribute to food and macronutrient intake? So taking a step back and first addressing the evolutionary theory behind body mass index, I'm going to read you this quote. So, for survival, mechanisms must exist to preserve the leanness and agility of the hunter, or in different circumstances, well-padded energy reserves to withstand famine. So, and Ethan Sims was a physician in like, I don't know, the 80s or something. But I think the point that I want to make here is that although the language is flowery, this idea that the prevailing theory is that of why most animals, including humans, have relatively consistent body mass. It sums up why people think humans and other animals have a pretty consistent body mass across the population. So what does this mean in, like, I like to put things in graphical form. So it means that over time, our bodies have mechanisms for making sure that we don't get too skinny so that we can survive periods of scarcity, and also that there are mechanisms to make sure that we can also don't get too heavy so we can escape predators and catch prey. However, humans in our, and let me tell you, sorry. So it's pretty well accepted that this compensatory measure for keeping us not too skinny includes the leptin melanocortin pathway, and that there is a catabolic signal of overfeeding that's theoretically keeps us from gaining too much weight. So the big catch in this, however, is that humans and our most recent ancestors have had very few predators and little trouble catching prey since we started using fire around 1.8 million years ago. And as the theory goes, this catabolic signal has undergone genetic drift or random mutations, not subject to selective pressure. So is the theory backed up by experiments? So the short answer is yes. In the interest of time, I'm not going to go into the particulars of this mouse study, but the authors did a really good job of demonstrating that this catabolic signal of overfeeding exists in the blood and that it works independently of the leptin melanocortin pathway. However, exactly what that blood-borne factor is still a mystery, and you can imagine it would be even harder to pin down in humans because of the recent genetic drift. So this is sort of my theory, but it's just a theory that potentially, like FGF21 and the pathway that it works in, which is only very preliminary, being described in molecular studies, could play a role in this as this sort of catabolic signal of overfeeding. So the reason I think that is that the genetic variants in FGF21 are associated with protein intake. This endocrine hormone is released from the liver. It's induced in the circulation by protein dilution, so if you have a diet that's very low in protein, and alcohol intake. And it acts on the FGF1 receptor, beta-klotho co-receptor in the brain. And Currently, there are analogs in clinical trials for food intake, and that reduce food intake, decrease body weight, improve lipids, and thankfully, like so far, there's very few side effects aside from diarrhea, and very few CNS side effects, which is predominantly the problem with other obesity drugs. So in sort of full here, my takeaway has been that it's probably safe to assume that that genetic variation contributes to macronutrient and food intake to some meaningful degree in humans, but to what degree is sort of unknown at this time. Okay, so now I want to know, how does this genetic variation in food intake relate to food access? So even though there has been genetic drift over the last 1.8 million years, humans have only grown more obese in the last 50 years or so. And importantly, only 1/3 of them have become obese, not everyone. So what genetic theory can genetic theory help us with this too? So without access to a Western diet, and I'm like not defining this here, so I know, people stay at a very tight range just above this lower intervention point. And this can be seen by looking at the very tight range of BMI in today's hunter-gatherer and subsistence agricultural communities, which all are around 20-ish. Yeah, from different parts of the world. Super interesting. Okay. So, however, with the access to a Western diet, which I realize I haven't defined here, 1/3 of humans gain weight over time. So why do some people gain weight faster, or younger, I guess you could say? Could genetic variation in the reward system in response to food intake be the reason behind this? So there are more than 20 regulatory gut and adipose hormones that have a wide variety of functions related to food intake. And a number of reviews, really nice reviews, have pointed to the potential relationships between some of these food intake hormones and chemical signaling pathways in the brain. Most attention is usually given to the endocrine effects on the mu-opioid pathways and the dopamine pathways, since they have been extensively studied in animal models of reward system deficits, such as substance use and addiction. We have evidence that these two pathways are relevant in obesity control in humans too, as a combination of naltrexone and bupropion are currently used to treat obesity, though there is much room for success with that treatment. However, the main takeaway is that while there has been substantial and provocative work in this area, no one has really systematically studied on a human population scale whether a genetic correlation exists between food intake reward and other reward system deficits. So this group, however, decided to take the idea of food addiction and just run with it. Instead of measuring the genetic correlation between food intake and reward system deficits, this group tested for the association of common genetic variants and a food addiction score. And they basically did this by adapting the substance use disorder criteria and surveys to and just like sort of substitute like food. I mean, it was a little more involved than that, but they basically like if there's any food where you sort of have like the feelings of craving, etc., they would like rate you, and then they looked at your genetics genome-wide. They looked at the genetics genome-wide of all of the participants. Okay, so yeah, I'm not gonna. So they said that they had found two that were that reached genome-wide significance, and they were neurotrimum and protein kinase C alpha. They said in their results and conclusions that they had no obvious roles in eating behavior. The only thing I could find in the literature was that that neurotrimum, they said was like there was something that said that it was it was a closely related family member to the opioid binding protein, which is potentially interesting. And that protein kinase C alpha is linked to memory capacity in healthy subjects and to risk of PTSD in genocide survivors, which was like really random, but interesting. Okay, so there's sort of this like, is addiction really appropriate here? So I'm going to read you this. This was like a commentary in an endocrinology Nature review. So the concept of addiction is loaded with connotations and is often used for its political as much as its medical utility. The hedonic or reward system can account for the ingestion of foods and drugs, confirming that they share neural substrates that differentiate liking and wanting. These are normal processes that are recruited for natural homeostatic behaviors, and it can explain the phenomenon of hedonic overeating as a consequence of human motivation pushed to extremes by an obesogenic environment. So I don't know, I just thought this was a really good reflection on before how to sort of study these concepts and in a way that really helps us understand the biology, but sort of being aware that this research is done not in a vacuum, and that there's a lot of is it really appropriate to be using the term addiction in this context? So then I want to ask, what actually is addiction? So from the American Psychiatric Association, the DSM, their definition is that addiction is a complex condition, a brain disease that is manifested by compulsive substance use despite harmful consequence. So they couldn't really come down on whether it was a disease or a condition. And then as for whether it's the substance use, they've recently also in the DSM-5 included non-substance related disorders under the heading of substance related and addictive disorders. It's very confusing, but this is what it is right now. Yeah. Yeah. Oh, yeah. Thank you for clarifying that reasoning for me. It's awesome. Thanks. Yeah, I mean, I don't know. Yeah, like I said. Okay, so now I'm going to get to the part of the talk. What time is it? To the research part of the talk. So thankfully, I have some time, which is great. Okay, so the first research gap that I was interested in looking more into was of the 300 or more common genetic variants associated with obesity, which are mediated through high fat and or sugar intake. And I was interested in the high fat, high sugar intake because it was very simple, and I think it also can address this issue of whether or not palatability is a word that's sort of thrown around in this context, and whether or not that really has legs to stand on, I think is something I still need to think about more. So the data that I would use to answer this question is available through a great resource called the UK Biobank. It's a group of 500,000 individuals that the UK has collected data for, and it includes, from a dietary perspective, it includes 5 serial 24-hour recall, half computerized, half dietitian sort of helped surveys, and then it also has genotyping data for all of the participants. The 24-hour recall data is only for about 200,000 people, but that seems like an appropriate scale that could be useful in this context. And then the possible method, and I'm was tentative to even put this up because I'm not really sure yet, would be using structural equation modeling to get at the answer to this question. And then the limitations, a very strong limitation, which I've sort of already alluded to, is that I really like simplicity. So one measure for sort of a combination of high fat and or sugar and low protein intake may sort of sacrifice necessary complexity to sort of disentangle what's really happening. And I got sort of interested in this geometric framework for nutrition. So that was one idea that is sort of percolating in my head. And then the second research gap that I was thinking about was that hopefully is sort of obvious to people from what I've discussed today, is that What is the genetic correlation between high fat and or high sugar intake and other reward system deficits? And again, the UK Biobank also has data on alcohol intake, tobacco use, cannabis use, and then sadly there are data available on other substance uses, but they're not quite as easily coded in terms of like use for me, but it's not impossible to use. And then some possible methods that I just sort of would be like the most basic would be an LD score regression or a polygenic risk score association. But these methods have serious drawbacks because of just like confounding. So they're just pure correlation. So there could be reasons why like somebody could be they're eating a lot because they have the munchies a lot. Like that's not really going to be very useful. And so, but the idea would be that would look something like this, where you could sort of evaluate, like on a very rough scale, like is there any genetic correlation between food intake and of high fat or high sugar intake and any of these other substance uses. And then the last gap that I was thinking about was what are the relative contributions of food access and common genetic variants in reward pathways on food intake, assuming they are actually causally related. Like, can we actually quantify the relative contribution? And so, again, like I said, I'm kind of like gung-ho on the UK Biobank, but I could also use food composition databases and price databases, or like in this case, it would be probably more appropriate to use the UK equivalent to look at this. And some possible methods up there, again, very preliminary. But like obviously, because I'm coming in with the assumption that I think that that those associations are really true, the burden would be on me to say like the assumptions that I decide may be invalid and may be less important to the community. So yeah. So that's the end of my talk, but I would really like, since there is time, to get your feedback and like what things seem like wishy-washy or that you think are really exciting and what you would want to know sort of in this area. So thank you.
First of all, I'm really interested in this, so that's a great talk. Thank you. But you seem to be relying on structural equation modeling as your proposed methodology. Okay, which means that all of a sudden you've got a limitation on the number of confounders you can look at. How are you going to screen for that? And two, just as a question, how is our mental health indicators going to come into this? Because if you remember Katie, being mentally ill is a very, very high predictor of obesity. So,
great question. Okay, so the first question was about confounders. So yeah, I think let me go back to this. So there's sort of this balance, right? Like these like very agnostic approaches are just like, oh, as association, you're sort of limited by like you could do model adjustment for making sure that like you add some variables that you think are important, but like really you're going to have this problem of confounding. And like if you make the leap to doing structural equation modeling, and I may be speaking out of turn here, so I'm just going to say that that like being really thoughtful about like what things from the literature really we think would be very important in terms of like choosing which confounders to include. I mean, obviously, like there's no like perfect way to do that, and it would be like I'd obviously have to like do a serious literature review to make sure that like whatever I choose can like stand on its own and against like the community. Oh, and then your second question. Sorry, and then I know people, so I know people have, so I looked at people who did cardiovascular disease, but not obesity. And then as to the mental health question, yes. I mean, the time issue is paramount, right? Like if we had, you know, time course data, like that would be ideal. And in this case, like, you know, what came first, the chicken or the egg, right? Like, how does mental health affect both of these things? Like, you know, it's a loop, right? In thinking about this problem, I feel like ignoring it is not good, but just sort of like throwing up your hands and saying like, oh, well, it's a loop, so we'll never figure it out. I don't know. I'm still like weighing that and figuring out like whether or not I think that it will be helpful. So this cohort does not include children. It does include their weight percentile at age 10, I think. It's either 10 or 8. So that's like something that could be helpful.
So, Kristen, this is awesome. I want to just dive into one very basic piece of this for a second, which is how are you envisioning doing the actual phenotypic classification, right? I mean, are you say high fat, sugar intake, and you also talk about obesity, right, as your like main outcomes and main inputs here. And fundamentally, how do you envision actually getting a handle on that? Yeah.
Yeah, I've thought so much about this, and I still feel like I don't have it like 100% like, oh yes, this is the way I should do it. And I think like as my questions get more like clear, I'll have a better like sense of like how exactly, what measures I'm actually going to use. I think in terms of like asking the question about food intake and its potential genetic overlap with various like reward deficits, I think that BMI is a better marker than say something like a better marker to use because we know that it's more like centrally regulated versus some of the like fat distribution outcomes that are.
But even for something like BMI, right? Like, are you going to draw a hard cutoff at something, or are you going?
I was going to use it as a quantitative trait.
You're going to use it as a quantitative trait.
Yeah, that was my like plan. But like I said, I'm not 100% sure yet.
So I remember last time we were talking about this. I don't think there's a button on it.
No, we're using that one.
The last time we were talking about this, we were talking about the gold standard distributions.
Yeah.
And whether or not you could leverage the gold standard measurements to arrive at a better power.
Yeah, I have thought about this a lot more too, and there was like a really nice paper done like a long time ago, I want to say like 4 years ago. Actually, the person who invented, let's see, okay. So the person who did Ben's index, right, with like the N there.
Right, that's what I'm asking.
Yeah. So like theoretically, right, you could like fit your, like given your population that you end up using, you could try to like fit the curve exactly to your population. There, my Occam's razor tendency sort of like slipped in of like, yeah, you could do that, but like.
And then the language you would have to use.
Yeah, and like it's mostly used in children. So like there's this like balance of like, well, are people gonna be like, why the heck are you doing that? Or
is it a way to reduce noise in your data set?
Yeah.
Where your outcome variable better matches what you're
So
doing.
what's interesting too is that on a subset of this population, they do have some of these other more rigorous or more gold standard methods. They don't have this method. I think they have bioelectrical impedance analysis for a subset of people. So yeah, I have an inclination to stick with BMI just because I feel like it's easily interpretable, and especially like if I am going to do something like that's, already kind of like confusing by including like confounders and stuff. Like, I want it to be more easily interpretable. So I'm not like sure whether I would go that route, but it might be interesting, at least for me in my analysis, to look at it. Like,
well, if you can justify doing the calculation this way, it might provide you with a clearer outcome.
yeah, I agree.
Thank you. From a pediatric perspective, there's a lot of interest in food insecurity, and I imagine this kind of ties into the stress pathways, and I had to go back and look and remember that you've got the leptin pathway is related to that stress pathway, but I just wanted to bring that up, and maybe, I don't know if anybody's done any genetic correlations or anything like that.
With just the leptin pathway, are you asking?
No, with response to food insecurity as regards to any, genetic mediator. But I think it's also important in your modeling as you make your primary
like stress,
assumptions.
Like, yeah, right.
Yes, but how likely it is that you'll have food next month is different from how much food do you have in total on average.
Yeah.
And that's an important factor, and it has a lot of policy implications too, because people simplify it and say, hey, well, they're just eating too much, right? So that and the stress response is probably a big mediator of that. I just wanted to bring that up.
Yeah, I mean, and that's like, so I brought up this concept of the reward system, and I'm like tentative to say too much about it because I'm not I didn't come from behavioral neuroscience training program, but I've learned a lot over the last few months, and the idea of liking, right, and whether or not liking in terms of reward is actually is it always liking, or is it just relief from instead of being this feels good, does it just feel not bad? And so if you do have that elevated stress response, what we call the reward pathway, I feel like it has to involve the adrenal system and stress.
And your threshold for all that stuff is probably changing as your stress changes. Yeah. And I wanted to bring up also in relation to that also, have you considered epigenetics in terms of so you've got poverty over 6 generations, and so you got DNA methylation and so on.
Yeah. So that was actually epigenetics was what of obesity brought me to this. But at this time, I still feel like there's enough unknown about just the genetics to really to then add the added layer of epigenetic regulation on top of it. I think at this population scale, it's a little preemptive right now, but absolutely, I would love to work on something like that in the future.
Thank you.
Yeah.
So you've got a lot of questions which kind of went into the methodology, and in the symposium it's more, just to remind people, versus the proposal defense. Yeah. It's much more about the gaps, which I'm glad you focused on. I do think, though, that it seemed like you were really driven by the data set, because UK Biobank came up in every possible question. And for some of the issues that you brought up, with Dion's question, one of the things you really have to think about for that data set is what is the clinical phenotyping. The mental illness piece is actually huge.
Oh, yeah, so it's all self-reported.
I know it's also, but that's what I'm saying.
Yeah.
So you have to really think about that. Potentially, if you had things around self-reported meds or something, you might be able to do a little bit more forensics, but that's a real concern. There has been extensive phenotyping in model organisms, especially coming from a behavioral neuroscience perspective, which would get around that. So you thinking a little bit more about how to leverage that data, and it doesn't mean you don't get to use UK Biobank, but I think I would come at it much more from the perspective of how can you tackle these challenges in a creative way that hasn't been done before.
Yeah.
And then ask yourself, what are the data sets or the things that are there, because some of that might help you get around some of these issues, at least kind of to bootstrap up into the very specific questions you want to ask, especially for the structural equation modeling. Dion's point was essential. You have to really think about what are the different components that are going to be in that model. And so you being able to leverage other data sets to help get there would be something to really think about as you're working on your proposal.
Thank you.
So,
Eric?
yeah, just on that point, should I use the microphone? Yeah. Okay. I'm very loud. Okay. So just on that point, I just wanted to latch on to that. So in the case of structural equation modeling, would having knowledge of priors in terms of what might be good targets be useful? And could you scour the neuroscience literature? Because we have a lot on using drugs to cause behavior as well as knockouts to affect behavior. And could you potentially use that literature as a way to kind of refine, what you're looking for in terms of what might make sense as either for priors or as a post-hoc sanity check on your outputs, since we do know a lot about that.
I think the idea of like doing like the genome-wide component when trying to combine these is not like that was not my intention. I think that's probably not like a good idea. Well, but that's my like immediate response. But I do think like using like animal models and even like having more of a like variants that are in specific pathways could be like a good way to get around.
Well, so your third that's loud. Your third aim is really is about causal relationships, right? So just out of curiosity, are you
MT2, can you just repeat your question? I'm sorry, Eric.
I'm worried about using one of those things. So one of the things that's coming up is this causal, is trying to identify causal relationships. And like I said, there's been a lot of drug studies, so I'm wondering if you've taken a look into the MT2 literature on both fat intake, sugar intake, and physical activity, which has been done extensively in rodents and to some extent actually in non-human primates like rhesus macaques as well.
Can you say MT2?
MT2.
I'm saying, can you say what that stands for?
Oh, I can't pronounce it. Like, was it
Is it like an actor?
No, it's a melanocortin.
Oh, okay, yeah. That's what. Okay. Yes. Okay.
So I don't remember what it might just stand for that. I don't remember at the top of my head.
Yeah, I think that would be like a very good way. Like, I mean, just like thinking off the top of my head, like it'd be a good way to like sort of like narrow down my like scope as to which, if I'm going to test things that have a lot of prior information in the literature, that would be like a good resource to use as like, so I'm not doing just like, it could be anything sort of search. I agree with you. Thank you. It's fine.
So if you're going to use animal models from the literature as priors, one could also think about maybe using human addiction pathways or something that are well known in the literature as priors.
Yeah, that was. Yeah. Okay, so Josh just said that while you could also use animal model priors that are associated with reward system deficits, that you could also, if there are annotated pathways available in any publicly available resources, that could be a good way to also narrow my scope as to like variants that are enriched in those certain pathways. Yeah. I agree.
So what would
Oh, sorry. Yeah. Thank you, everyone. I really appreciate your comments.
People should feel comfortable to ask your questions. It's just that we're out of time for the room.
Yeah, please come talk to me. I'm chilling.
Happy to chat later.
Yeah,
We'll
I'll
both
love
catch
you.
up at some point.
It might actually be called like Melanocortin 2 or something stupid like that. So we used to try to look at effects on social behavior in rhesus macaques while at parakeets because we were interested in effects in the reward system.
So can you say it again?
MT2 has direct effects on the reward system, particularly through melanocortin receptors, which also affect oxytocin signaling as well.
So good. That's all I have to say. So good. Loved it. I learned so much.
They numeralize it usually, so it's usually an II for some reason, even though it's 2. It's so stupid.
It was awesome.
It's fine. It's fine.
So if you give rodents MT2, it completely affects their food intake dramatically.
Yeah, dopamine knockout. No, did I take this off?